And then I am going to record it. Are you okay with that?
This is being recorded. Can I say got it? Yeah. All right. So Ty has been in my lab. I've been working with him for almost a decade. And actually only just a couple days ago, despite having worked with him for such a long time, he just told me that you pronounce his last name Chiaro. So this is Ty Chiaro's defense. I've said Chiaro for 10 years, so embarrassing. So I don't know if the new graduate students are here, but don't get scared because it doesn't take 10 years to graduate with your PhD. As many of you know, Ty joined my lab as a master's student. So over these 10 years, I've gotten to know Ty very well, and it's been a pleasure to mentor him. And I could go on and on and on about all of the different stories about Ty, but I've decided to highlight 3 qualities about Ty that have impressed me over the years, that have developed over the years, and that I think have led to his success as a graduate student and will lead to his continued success. So the first one, oops, no advancement. I don't know why it's not working, but I'll let you figure that out. The first one is his work ethic. So I asked the lab for pictures, and everyone sends me pictures of him sleeping in the lab. From day one, when Ty joined my lab as a master's student, you don't get paid as a master's student. You have to work a job to pay your rent and feed yourself. So he worked a full-time job at ARUP. And instead of just working, here and there in the lab, he worked every day. He worked as many hours as the other PhD students in the lab. So from day one, he impressed me with his work ethic. He continued that throughout his PhD, and he continues that now, a time when many graduate students kind of slack off, they're finishing things up. He's in the lab, he's working really, really hard. The amount of hours he puts in it impresses me, and he's had to do it because he's had to try so many different things throughout his PhD. But he also reads a lot. He reads very deeply, so he spends a lot of time doing that as well. And then he applies his work ethic to helping others in the lab. So many commented on how often Ty has helped them either intellectually or physically with an experiment. So I think he's sleeping in the lab not because he's lazy, rather he works so many hours in the lab that he's not sleeping enough at home, so he has to take little naps when he's in the lab. So the second thing that really impresses me about Ty is, and that has developed over time, is he initially came in with a very strong scientific curiosity. I think that's what brought him to my lab in the first place for his master's degree. But what's really developed over his PhD is his innovativeness, is his creativity, if you will. And scientific curiosity often leads to innovativeness. And I think you'll see this when he presents his unpublished work today, is how innovative he's been. The work he's going to talk about today was not my idea. It was not driven by me. In fact, I wasn't very excited when he decided to take this path. But it's turned out to be a really important finding in the field, a really fantastic story, I think, and really driven by Ty's scientific curiosity. He is incredibly animated when he talks about something. So I tried, this picture is him being animated. And this other picture is we have an innovation roundtable every year. We haven't done it in the last couple of years, but he teamed up with Warren one year to present his innovative ideas. I wrote this so I didn't forget. In the last couple of years, Ty has come into my office during our individual meetings, and I swear every time he starts with, "You're going to hate this." And at one point, I did hate the things that he was proposing, but over the last 2 years or so, I have loved them. 3 years, 4 years, whatever it's been. Every time he starts with that, "You're going to hate this," I end up loving it. Whether it be an experiment, a direction. More recently, it's been secret experiments that he's conducted in the lab, and he's showing me the results. Again, scientific curiosity and innovation have really been a strength of Ty's. The last one has been grit, or resilience, if you will. Again, Ty has had this from day one, but it's really developed over time. It's something that's, I think, very important as a scientist, because there's a lot of failure, a lot of rejection. And Ty had this early on. He published a really important paper in Science Translational Medicine early in his PhD career. It wasn't easy getting that paper in. It was looking at fungi in the gut at a time when people, first of all, didn't think fungi were really present in the microbiota, and they really didn't think that they were going to do anything relevant. So getting that paper published took a long, long time. And Ty persisted. He worked really hard to do those experiments. Once that paper was published, we even had a person write to the editorial board about it, about their kind of critiques, and they didn't necessarily believe all of the things in it. So again, at an early stage in his PhD career, he had to convince the editor that to not publish this critique that they had put forth. Now, of course, when you publish an important paper early in your PhD career, it's hard to know which directions to take it to continue your PhD. Ty took a lot of different paths. I think it was represented by his thesis committee. We had to ask people to come on with expertise. He tried to go in the autophagy direction. We worked with Eric Schmitz to isolate a molecule that was being secreted by fungi. He went in so many different paths. And again, he never got down. He just kept persisting. He just has like that grit, and you really need it to be a good scientist, and Ty possesses that quality. So all of these things have led to his successes in the lab, which I've already mentioned, his first author science translational medicine paper. He's written several commentaries in the field that have been published in Science and Cell Host and Microbe. He's currently writing up a review, an invited review that's going to be published in Nature Gastroenterology. And then, of course, he's going to present the manuscript that we're putting on the finishing touches on, again, which I think is a really important contribution to the field. He has helped many people in the lab, so he's had a lot of secondary authorships, including papers that have been published in Cell Reports and Nature. And I wanted to highlight like how important Ty has been just for my career development, because he has been in my lab as long as I've had a lab. And all of the experiments that he did served as the preliminary data for my first NIH R01, which is like a huge thing when you have your own lab, is you've got to have that NIH R01. He also did all the experiments that served for the preliminary data for a foundational grant in the Crohn's and Colitis Foundation. So he's been really important just for the lab, foundational member of the lab. And I wanted to point out that he really has began two different areas in my lab. You don't think graduate students are going to come up with these areas, but I didn't work on fungi in the gut before Ty came, and now it is a major focus in my lab. The experiments that he'll present to you today also represents a completely new direction that I wouldn't have thought to come up in, and that's looking at how innate immune receptors influence the microbiota composition, an area that we've just recruited a graduate student to carry on in lab. So very, very important for the future direction of my lab as well, and his legacy will live on. I wanted to also point out that his successes have garnered him a really prestigious postdoctoral fellowship in clinical immunology at UNC. There are 3 of these programs in the country, UNC being one of the better ones, and they only take 1 person every 3 years. And Ty was the person that they picked this year. So this is something that is just really an amazing accomplishment for him. So it has been my pleasure to work with Ty for such a long time. And with that, I will turn the podium over.
Let's see if we can get this to work. Thank you, June. Really appreciate everybody being here. Thank you to family and friends. And thank you for the department for being here. I hope to get this right for you. Okay, I would like to start off by dedicating this presentation to my best friend, Ricky Ford. And I don't think anything can really represent him and I's relationship more than this picture right here. But whether we were on the football field or the wrestling mat or just being ridiculous, Ricky and I were always together. And they tell you when you enter graduate school that life's gonna move on and things are going to change, but you don't really know what that means until you lose your friend. And so I just wanted to honor him and dedicate this presentation to him, because he was honestly one of the most fearless and courageous individuals that I ever knew. And it's interesting that June talks about grit, because I feel like when graduate school got hard, I often look back to those times on the football field or the wrestling mat, and I recall how freaking tough he was. And I really hope he's going to be with me today. I think he will. Now, when you study the gut, you tend to come across some interesting factoids. And some of these factoids tend to be pretty obvious, but no one really articulates them for you until someone like myself says it out loud. And so one of these factoids that I came across was, average lifespan is 80 years, plus or minus, right? You can go your whole life and never have sex, and you can live a complete life, no problem, right? But if you didn't go to the bathroom, particularly number 2, for 2 weeks, you'd be dead. I think this really highlights the importance of the gastrointestinal tract and how important it is to maintain tolerance to the microorganisms that live within the gastrointestinal tract, and how important this system is to our well-being. Now, I'm going to be talking today about inflammatory bowel disease, and you can really break inflammatory bowel disease up into these 4 epidemiological stages. And now, 20 years ago, people might have said, well, IBD is a problem of the Western world. And that was true, and it still is mostly true, because it was the late 18th century that we very first started to see the emergence of this disease, which is the first sporadic cases of IBD that pop up in a population. By the mid 20th century, we started to see cases within the newly industrialized countries. And now in the 21st century, we are starting to see the emergence of IBD in developing countries. Next comes the acceleration in incidence, where the incidence begins a steady incline, but the prevalence remains relatively low. Next is the compounding prevalence, where the incidence actually starts to level off, but the prevalence really starts to skyrocket. And lastly is the prevalence equilibrium, which is really a theoretical framework in which the incidence actually starts to taper off or even decline, and the prevalence declines or comes into equilibrium due to mortality beginning to be approximated. Now, this is really beginning to be a global problem. As you can see, in the last 30 years, IBD has increased in the last by 85% worldwide. And you can appreciate that North America, the United States, Australia, and Western Europe still have the highest compounding prevalence, but we're starting to see higher and higher cases within Asia, South America, and Central America. And the emergence is now beginning in more developing countries within Africa. It's thought that 7 million people worldwide suffer from this disease. And by 2030, 1% of the total population in the United States will have some form of inflammatory bowel disease. Now, this is not a homogeneous disease. You can break this into 2 distinct diseases, ulcerative colitis and Crohn's disease. Ulcerative colitis only affects the colon. It is continuous in its presentation, and it affects only the innermost layer of the tissue. Conversely, Crohn's disease can affect any portion of the gastrointestinal tract from top to bottom, and it tends to present in patches. So you can have diseased or inflamed tissue right next to healthy tissue. And this can spread through all the layers of the tissue that often have to be repaired surgically. Now, IBD is a complex disease, and the etiology is not well understood. It is thought to develop from a complex interaction between the environment and our genes, okay? And things like smoking, your diet, whether you had a gastrointestinal infection as a child or took a lot of antibiotics, and there's certain mutations that have been associated. But regardless, your environment, the things that you eat, the things that you subject yourselves to, and your genes will all play a role in the composition of the microbes that live within your gastrointestinal tract, and we call them the microbiota. As the microbiota changes, this can drive inflammation in conjunction with genes. This can lead to an overall dysbiosis, which can further feed back into more inflammation, really representing a chronic inflammatory state that we see in IBD. Now, most of the organisms that we study and that have been studied in the context of IBD are prokaryotes. They're bacteria. But as June mentioned, there's several different species of fungi that live within the gastrointestinal tract. One of them, Saccharomyces cerevisiae and Candida albicans. And some others, Malassezia and Penicillium, are known to live within the GI tract. And when I very first came into the lab, June alluded that there was a project that had just barely begun, and it was based upon a paper that had recently come out that was one of the first to identify differential fungi in the guts of patients with IBD. And we acquired 2 of the different fungi from that paper, and we performed some colitis experiments. So Saccharomyces cerevisiae, which was only present in human stool and was not found in any of the IBD patients, only healthy controls. In comparison, Rhodotorula aurantiaca was found in both Crohn's disease and ulcerative colitis patients. So we thought, great, we've got a negative, we've got a positive, we're going to do our colitis models and let's see what happens. Surprisingly, over 2 different models of colitis, Saccharomyces cerevisiae exacerbated disease, as demonstrated here by the higher colitis score, whereas Rhodotorula really didn't make that much of a difference. Now, I did a lot of different experiments. We did some sequencing, we did some metabolomic studies, and ultimately we identified that purine metabolism was that fungi could differentially influence purine metabolism on the host. And the final product of purine metabolism, you might recognize uric acid. It's the etiological agent of gout, and it is oxidized from hypoxanthine to xanthine by this enzyme xanthine oxidase. And we thought that maybe if we could inhibit uric acid production, perhaps we could ameliorate disease. So we use this drug allopurinol, which is a competitive inhibitor of xanthine oxidase. And what we were able to do is actually ameliorate disease in our colitis model when animals were orally given Saccharomyces cerevisiae and allopurinol. This was one of the first studies to really functionally test different fungi in the context of inflammation, and one of the first to really look at the metabolic output and impact of fungi on the host. Now, as June mentioned, I went in a lot of different directions. I really tried to tease this thing apart as to how the yeasts were driving this uric acid metabolism. I hit my head on the wall for a long time, and ultimately I wasn't really able to figure it out. But we did kind of hone in on uric acid and some of the consequences of uric acid. So uric acid is primarily made in the liver, and it's excreted through the kidneys, mainly 2/3 of it is secreted through the kidneys, 1/3 of it into the intestines. But there's another way uric acid can be made, and that's when cells die, whether they die inflammatorily via necrosis or pyroptosis or apoptosis. Xanthine oxidase can be released from these cells and continue to oxidize substrates in the cellular milieu, ultimately leading to the production of uric acid. Now, uric acid has been described as a damage-associated molecular pattern that can make other cells in the surrounding area aware that something bad is happening. One of the receptors that's been described to bind uric acid is CLEC12A, and this is the receptor we're going to be talking a lot about today. This is a C-type lectin receptor that has an immunotyrosine inhibitory motif on the intracellular end, capable of recruiting SHP1 and SHP2 and limiting inflammation coming from other C-type lectin receptors or toll-like receptors. So really, I've always called this thing a brake. And if you think about what a brake does in your car, you don't just come to a stop immediately, but you gradually slow down. And I think about CLEC12A as constantly surveying the environment and able to pump the brakes a little bit on an inflammatory event. Now, CLEC12A is primarily expressed within the innate immune system and only expressed in the immune system. We don't see it in epithelial cells. And I have looked at innate and adaptive immune cells within the GI tract, particularly the colonic lamina propria. And that's what I see. I see expression within neutrophils, monocytes, macrophages, and dendritic cells with very little to no expression on lymphocytes. And when we look at the expression on a per cell basis, we see that neutrophils have the highest expression of CLEC12A, followed by macrophages and dendritic cells. CD4 T cells, little to no expression, with just a little bit on CD8 T cells, which is consistent with what other people have found as well. Now, I told you that there is a genetic and immunological component to the development of IBD. And one of those genes is this gene right here, ATG16L1. And ATG16L1 operates within the autophagy pathway, and it's important for sensing cellular stress and responding to invading bacteria. However, people with Crohn's disease have a mutation in this protein that makes them susceptible to caspase-3 activation. If they have this mutation, caspase-3 cleaves this protein, and ATG16L is no longer able to respond to cellular stress or an invading organism. So this next group took peripheral blood mononuclear cells from patients with this Crohn's disease mutation and stimulated them with different bacterial ligands, and then performed RNA sequencing. And what they found was CLEC12A was the most downregulated gene of these mononuclear cells stimulated with bacterial ligands. So given our interest in uric acid and this receptor's importance to inflammatory bowel disease, we really wanted to understand how CLEC12A maintains intestinal homeostasis. Sorry, one sec. So we started out with a colitis model. We acquired wild type and CLEC12A knockout animals, which we thought were relevant since this gene is significantly downregulated in human patients. And we performed a 7-day DSS or dextrans sodium sulfate experiment where there was provided in their water. And we're going to monitor weight loss and colon length as a disease readout for these animals. And what we saw was that in the red, CLEC12A knockout animals lost significantly more weight over time and had significantly shorter colons. Now, we're going to see these graphs a lot today. So to make it very simple, more weight loss is bad, shorter colons, bad, okay? Again, we're going to see this all day. So we can really begin to build this model whereby CLEC12A, in the context of some type of intestinal insult, be it colitis, changes in the microbiota, a pathogen, really it acts to limit inflammatory signaling, okay? And so from here, I really began to wonder, well, which of these cells is really responsible for driving this exacerbated colitis phenotype? And then beyond that, how does CLEC12A really impact the immune system in the gut? Now, surprisingly, and not surprisingly, but it's a relatively newly discovered receptor. There hasn't been that much research done on it yet. And actually, one of the only studies to look at CLEC12A has been in the context of a secondary lymphoid organ, and that's the spleen, where they said no changes in monocytes, neutrophils, eosinophils, et cetera, no changes in the adaptive immune system, done, right? And I just thought, okay, well, let's actually look inside of a tissue and see what the real story is. And one of the things that I noticed early on within the colonic lamina propria was that these animals were significantly deficient in neutrophils within the tissue. The other thing that I noticed was there was a significant expansion of T regulatory cells. However, these T regulatory cells were deficient in their ability to secrete IL-10. And we also saw a significant expansion of Th17 cells. Now, although CLEC12A is only expressed within the innate immune system is very important for activating the adaptive immune system. And the first question I wanted to ask was this adaptive immune system required for exacerbated phenotype? In order to do this, we took our CLEC12A knockout animals and bred them onto a RAG background. And then we performed our same DSS experiment as before. And what we saw in the blue and in the red, RAG CLEC12A knockout animals or CLEC12 knockout animals alone lost the same amount of weight as they did before. And they had equally short colons, indicating the same level of disease. And this really suggested to us that B and T cells weren't required in the CLEC12A knockout animal for the exacerbation of disease. But what about these neutrophils? I had told you that neutrophils were significantly reduced at baseline, but during disease, they were actually significantly upregulated. And so we thought perhaps neutrophils were the driving cell type for this phenotype, and we wanted to study that further. In order to answer that, we again kept the innate immune system isolated by using our CLEC12A and RAG double knockout animals. We administered an anti-Ly6G antibody that depletes neutrophils on days -1 and 4, and then carried out our same DSS experiment. And what we saw was that when CLEC12A knockout animals received isotype or the depleting antibody, they lost the same amount of weight. It didn't make a difference. Equally, they had shorter colons. So this really told us that we know B and T cells don't matter. Neutrophils weren't driving disease. But what about these macrophages? We know that the gut is constantly replenished by monocytes exiting the bone marrow, extravasating into tissue, becoming macrophages. And in the tissue, they can take on a spectrum of roles. We tend to delineate them as M1 and M2, which is silly, but bear with me, it makes it easy. M1 macrophages being inflammatory, M2 being more reparative. And I noticed in my double knockout animals that there was a significant expansion in the tissue of these M1 macrophages during disease. In the single knockout animals, I noticed the same significant expansion. And also there seemed to be this absolute lack of M2 or reparative macrophages in the tissue. Furthermore, we performed a cytokine analysis on colon explant tissue from DSS animals, and we noticed that CLEC12A animals had a significant increase in MCP1 or CCL2, which is responsible for recruiting monocytes from the bone marrow to sites of inflammation. So we hypothesized perhaps it was these macrophages that were driving disease. Now, in order to answer this next question, we performed an adoptive transfer of wild type or knockout monocytes. We would transfer wild type monocytes into a knockout animal or knockout monocytes into a wild type animal. We would deliver these on days 4, 5, and 6. Animals would be subjected to DSS colitis for the first 5 days and then given 5 days to recover. And what we saw was that when knockout animals in red were given wild type monocytes in blue, by day 10, we started to see not quite significant, but a trend to increase in weight, suggesting recovery from disease. But what I actually thought was really interesting was that when a wild type animal in gray was given a knockout monocyte, they actually sagged below in their weight loss and weren't able to recover. And this is better seen here in colon length, where wild type animals given knockout monocytes actually have significantly shorter colons, meaning that the absence of CLEC12A alone is significant or is sufficient to drive disease. Also, when we give a knockout animal wild type monocytes, we're able to rescue that and improve the disease phenotype. And lastly, one more piece of evidence to convince you, this is lipocalin-2 in the feces, which is a non-specific marker of inflammation. And when we give knockout monocytes to a wild type animal, we see a trending increase. And when we give these wild type monocytes to a knockout animal, we're able to reduce the reduction, lipocalin-2 in these animals, really suggesting that CLEC12A is a powerful modulator of the immune system. So we can kind of round out our model here a little bit that during colitis, CLEC12A acts to sense damage and limit MCP1. It also limits other inflammatory cytokines like IL-6, TNF-alpha, and IL-12, and really promotes more of an M2 expansion. But this maybe isn't really that surprising, right? I mean, we removed a major immunological break from the system, and now these animals are overinflamed. Although I will say that at baseline, they're not overinflamed, okay? So I really began to wonder what's happening here at steady state in the healthy epithelia. I told you that there were changes in both the innate and the adaptive immune system, and CLEC12A has been implicated in participating within the phagocytic pathway. And so given this, I really began to wonder, what's the impact of the microbiota in these animals? And so we turned again to our DSS model, and the DSS model is actually really nice to study the microbiota because it's dependent on the microbiota. When we give DSS, this chemical actually erodes this layer of mucus in the intestines, which brings the microbiota into close contact with the epithelia. And this drives a lot of the initial inflammation. And so in order to study how different microbiotas might influence, this represents a great model. And so in order to do this, I administered antibiotics in the water of these animals for the first 7 days, and then subjected them to DSS colitis for the next 7 days. And what we saw was that wild type and CLEC12A knockout animals that received antibiotics had a significant improvement in their colon length, which suggests that it's not just the loss of CLEC12A. There's something about the microbiota that's sufficient to drive disease in these animals, and we really wanted to know what that was. Okay, now the physicians in my lab hate this, okay? But just bear with me. Maybe this will make them happy. These are broad spectrum antibiotics, okay? But I will argue that they have a bias towards gram negative or gram positive, all right? Gentamicin primarily inhibits gram negatives. Cephalothin primarily inhibits gram positives. We're just going to use this as a tool. We're trying to figure out which, if any, of these groups are responsible for the disease phenotype that we're seeing in our animals. So I conducted this the same way. We put the antibiotics in the drinking water. We subject animals to DSS. And what we saw is within knockout animals in the green, in the purple, are given gentamicin, we see no change in the weight loss here. okay? But when we give them cephalothin, we see a significant improvement in their weight loss. We also see a significant improvement in animals given cephalothin in their colon lengths, and we see no change when given gentamicin. So this really suggested to us that perhaps it's a gram-positive organism that's driving disease. Next, we want to know, is this microbiota as a whole really sufficient to drive disease? And if so, who are the players? So now, here's where things get fun. You guys have probably heard about fecal microbiota transfers in the literature and in popular culture. We do those a lot in our lab. And so I took wild type feces or collect-12A knockout feces, and I colonized wild type germ-free mice. So these animals are collect-12A competent, all right? We let these microbiotas homogenize for 4 weeks, at which point I'm going to take half of the animals in the wild type group and half of the animals in the knockout group and combine them into a co-housed group, and then subject all animals to DSS for 7 days. Now, the reason this works is because animals are coprophagic. And what that means is that they eat each other's turds. And when I say eat each other's turds, I don't mean they're just living amongst them in the cage. I mean, they pick them up and eat them like a candy bar, okay? It's true, I've watched them. This is a great way for the microbiota to be amalgamized within the host and for us to really be able to study which organisms are driving disease, especially when to avoid things like cage effect, which we know is a big problem in the microbiota field. And what we saw was that animals that received a collect-12A knockout FMT or who were co-housed, meaning animals that originally got a wild type FMT, but then lived and ate turds of a knockout FMT, lost significantly more weight over time. And they also had significantly shorter colons. There's one more piece of evidence. When we look at the colitis scores, we see that animals that received knockout FMT or who were co-housed had higher colitis scores, which really suggests that the microbiota alone is different in a collect-12A knockout animal, and it's more cholitogenic. Now, we've been using this collect-12A knockout animal, which is great. It's a whole body knockout. But sometimes these animals can have off-target effects on non-immune cells. And so I really wanted to isolate the hematopoietic compartment to see if the immune system alone was sufficient to drive the changing of the microbiota and ultimately disease. So in order to accomplish this, I took wild type mice and irradiated them, reconned these animals with either wild type or collect-12A knockout bone marrow, and then subjected them to DSS colitis. Now, these results weren't as striking. We saw that animals that received a knockout bone marrow did kind of sag below the wild type bone marrow, and they had trendingly shorter colons than what we'd seen before. But before I subjected these animals to DSS, I took feces from these animals and I monocolonized, not really mono, but I colonized wild type germ-free mice and performed the FMT experiment just as I've described before, created a co-house group, and then conducted DSS. And what we saw, again, maybe not quite as striking by percent weight loss, was that the co-house group trainingly lost more weight compared to the other groups, although there was no statistical difference between wild type and knockout. But we did see shorter colons in collect-12A knockout, bone marrow, FMT animals, and animals that were co-housed with the collect-12A bone marrow knockout animals, which really suggested to us that the immune system alone is sufficient to reshape this microbiota. Now, I told you that B and T cells don't matter for driving disease. But when we think about the factors that really drive the changes in the microbiota and lead to what I like to call sculpting, there's a significant gap in the field as to what are the contributing factors that really lead to this. When we talk about this, a lot of people mention IgA. And interestingly, IgA is dysregulated in these animals, okay? I see less IgA positive bacteria in the feces of collect-12A knockout animals. And I also see more free IgA in fecal washes of collect-12A knockout animals. And so we began to wonder, maybe these cells aren't required for the initial disease, but maybe they are required for this differential sculpting effect that I see. And so in order to accomplish this, we're going to do our same FMT experiment, only this time we're going to use rag feces or rag CLIC12A knockout feces. And what we saw is the same thing we saw before. Animals that received a rag double knockout FMT or who were co-housed with rag double knockout FMTs lost significantly more weight, and they had significantly shorter colons. So this tells us that B and T cells are not required for disease, and they're not required for the microbial shaping. And this was really an important indicator that CLIC12A is an innate immune modulator of the microbiota. And again, when I think about sculpting, I think about the greatest sculptor of all time, and that's Edward Scissorhands. And I think about this constant interaction with the microbiota that the innate immune system has. And I think about the special tools that Edward had in his hands that was able to recognize certain bugs and get rid of certain bugs, maybe without hurting anyone else, only creating a great product. And so now we really want to move into who are the players that are different in the microbiota. And in order to talk about who the players are, we have to have a little bit of a discussion on microbial ecology. And if you're anything like me, you probably have a look like this on your face. Because for the last 7 years, when I've talked to Zach Stevens in our lab, this is how I look at him when he talks to me about microbial ecology, all right? But I'm going to break it down real easy, okay? In the gut, we see a typical microbial distribution looking something like this. We have abundance on the y-axis and species on the x-axis. We see some organisms are highly abundant and some are not so abundant. Things are not equal in the microbiota, okay? We also have several different metrics of diversity that we classify as different. We have observed features, which is simply how many. We have evenness, which is what you're looking at here, which is not even, right? And then we have an overall statistical calculation that takes into account the first two to say, are these things different? So when we look at my alpha diversity metrics, we see that observed features are not different. There's no change between wild type and CLIC12A knockout animals. We do see that CLIC12A knockout animals are more even compared to wild type animals. And that really drives an overall difference in our Shannon entropy, suggesting that these microbiotas are different. What does that mean? Conceptually, what does that mean? This is what it means. A CLIC12A knockout animal looks more like this. This is bad because if these are good guys, all of a sudden they're not very abundant. And if these are bad guys, all of a sudden there's more of them. So a more even microbiota is not necessarily good. And in this case, we know it's not good because we can see the effect of disease. So to really look at who was different, we identified this organism, Faecalibacillum rodentum, which was significantly different in CLIC12A knockout animals when compared to wild type, and also significantly more abundant in CLIC12A knockout animals when compared to wild type. And so, interestingly enough, Faecalibacillum is gram-positive. And remember when we treated with an antibiotic that inhibited gram-positive organisms, we were able to ameliorate disease. So we really wanted to know, is Faecalibacillum able to induce disease in wild type animals? And so in order to do this, I would orally gavage 10 to the 7th CFUs of F. rodentum and then subject these animals to DSS. And what we saw was that animals that were received live or fixed F. rodentum lost significantly more weight over time and had significantly shorter colons. Interestingly, we also saw a significant expansion of those M1 macrophages, and we also saw an increase in Th17 cells, suggesting that perhaps F-rod or F. rodentum is a pathobiont, an organism that doesn't normally cause disease, but in the right microbial situation, under a particular amount of stress, can lead to an inflammatory event that we see here. But how is CLIC12A really doing this? I told you guys that CLIC12A was a receptor for uric acid, but the full story is a little bit more complicated than that. In 2008, it was actually reported that CLIC12A is an endogenous ligand or a receptor for an endogenous ligand that does not recognize bacteria. By 2014 is when it was shown to recognize uric acid and lead to an anti-inflammatory event. 2015, it's shown to participate in antibacterial autophagy and either directly or very closely associate with bacteria. And in 2019, it was actually shown to bind to the plasmodial product hemozoin, where it actually promotes inflammation. So the full story is not known. So I began to wonder, could CLEC12A actually recognize members of the microbiota? And could this be how it is differentially sculpting the different communities that we see in the microbiota? And so in order to answer this, we created a fusion protein that contained the extracellular portion or the binding portion of CLEC12A fused to a human IgG1 Fc portion of an antibody. And from here, we created a flow assay where we would use this like a primary antibody and incubate it with different bacterial cells, and then hit this complex with a secondary antibody that we could analyze via flow cytometry. And what we saw was that our protein construct here was able to abundantly bind to organisms like Aphrodentium and Roseburia species, both gram-positive, but not so much to other members of the microbiota, and not at all to C. albicans. So we can really kind of finally complete this model whereby CLEC12A recognizes particular members of the microbiota and really adds a little bit of precision that leads to the clearance and promotes an anti-inflammatory or regulatory event, while at the same time, while these organisms are being detected by other toll-like receptors or even C-type lectins, able to downregulate the inflammation there. So I'd just like to take a quick minute to just highlight my contributions, I think, to the field. And one of them is just yeast being important in the context of inflammatory bowel disease, and that purine metabolism is sufficient to exacerbate colitis. We've really identified CLEC12A as an innate immune sensor, potentially for gram-positive organisms that might be really important for this sculpting effect that we see. And we've identified Aphrodentium as a potentially novel pathobiont that it could be important for driving inflammatory responses in the gut. Now, at some point in this, I had this animal that seemed to be very inflamed under cholitogenic conditions. And I began to wonder, how am I going to use this to benefit anyone, right? Like the fact that this animal gets so inflamed, who cares? And I wanted to see if I couldn't do something to actually make this inflammation work for me. And there was a few different contexts in which I thought this might work, but the one I thought that was the most relevant to me and to our lab was colorectal cancer. And so I employed this model of MC38 colorectal cancer, where we inject these cancer cells into the hind flank of these animals and they develop tumors. And I just really wanted to see, could a CLEC12A knockout animal, because of its inflammation, actually promote a better immune response in the tumor? And what we saw was that CLEC12A knockout animals seem to be almost refractory to tumor development over time. And I think you can really appreciate that here in this wild type animal. You can see this tumor, which is several millimeters long and wide, and it's really even hard to see in this CLEC12A knockout animal. We saw expansions of Th1, Th17, and CD8 T cells producing interferon gamma, and they were all producing more of these cytokines on a per cell basis. So what do you think the next thing I did was? Of course. So I did my same FMT experiment, took wild type or knockout FMTs, colonized wild type germ-free mice, gave these mice 4 weeks to colonize, and then we performed our same MC38 model. And shockingly, this microbiota promoted less tumor development over time. We saw a trending decrease in the overall tumor mass at the end of the experiment. Not to quite of the same magnitude, but we did see a significant increase in Th1 cells and CD8 T cells expressing interferon gamma. And so really kind of highlights to me that this receptor is very powerful and has the ability, if we can learn how to modulate it, could really be used therapeutically, either for the amelioration of inflammation in something like colitis, or is there any way that we can ramp it up in order to maybe promote an anti-tumorigenic effect? So that is the end of the portion of the data for my talk, and I'm happy to take any questions. Oh, Dave, yeah.
And your experiment is really, really nice with the colorectal cancer cells. Can you add the ephrodentium to those animals and see whether that has effect?
I think that's a good experiment. So interestingly enough, there was a paper with ephrodentium in which it was able to inhibit tumor growth, but not via inflammation, via its production of short chain fatty acids, which actually inhibited histone deacetylases and prevented tumor growth. So I think it would be interesting to do in the context of the knockout animal to maybe use these synergistically together. Yeah. Will.
Hey, first of all, I really enjoyed that talk.
Thanks, buddy.
Yeah, and second, so CLEC-12A knockout really shifts everything towards this more pro-inflammatory program, and all the anti-inflammatory things are really down, right? And so in my mind, I really think that these anti-inflammatory things are also the same programs that drive things like wound healing, for example. So my question is, if you treat your animals with DSS, like what's the recovery like in these knockout animals compared to wild type animals?
So if you remember that monocyte transfer experiment, that was kind of a recovery model experiment. And without going all the way back through the slides, the knockout animals were still on a downward trend at day 10. Whereas wild type animals were starting to come back up. And recall that the wild type animals that had received those, knockout animals that had received those wild type monocytes were starting to recover. I was always kind of interested in a wound healing model to really take that out. And you always kick yourself after an experiment when you think you took it too soon. I would have really liked to take those animals 5 days later to see if we could have pulled apart some of those differences. Yeah.
Great, thanks.
Ervik.
Great talk.
Thank you.
And I have a question. Do you know what regulates CLEC-12A expression and whether microbes can play a role in regulating CLEC-12A?
I don't know, and I don't think it is known what regulates CLEC-12A expression. I do know that usually upon binding of its ligand, which to this point has been uric acid, it's known to be downregulated. I will say that I think CLEC-12A is important for regulating other toll-like receptors and C-type lectins. But to what's actually regulating its ability, I don't know. Matt Williams first.
So I know there's been kind of growing evidence that there's microbial products in tumors. So I'm kind of wondering if you think, for example, in this setting, you could think of a model where macrophages in a setting of the gut might migrate into a tumor and regulate response, or whether you think that local recognition of microbial products is really required for this CLEC-12A suppressive effect.
I don't think that microbial products are required for the suppressive effect. Interesting that you bring up microbes and tumors. We just had a very dense paper in Journal Club about this topic. I don't know that I have a great answer for you. This is obviously an ectopic model, on the hind flank, so it's not directly in the gut. I am a little bit of a skeptic when it comes to some of these things about, something trafficking from the gut to other sites, although I know that it's been shown for things like the thymus. I think it would be interesting to look at in total, but I don't know that I have a great answer for you. Matt Mulvey.
Yeah, I was, is there anything known about the bacterial ligand for CLEC-12A, either with salmonella or ephrodentium? Do you know what?
No, but I really want to do that experiment. I've been like, I'm like, as soon as this is done, I'm like back trying to figure that out. No, in the previous paper that looked at salmonella and antibacterial autophagy, they used some microscopy to say, oh, look, these two colors are close together, but they didn't do any biochemical analysis to see what the C-type lectin might actually be binding to. Little tidbit, the C-type lectin binding domain is actually more representative of a natural killer receptor. And so other people have postulated that it does have the potential to bind to a protein. So I think if that's possible, we might be able to resolve that with something like a denaturing gel. I really want to we've got some tools in the lab now with this construct and an IP assay to really maybe make some protein extracts from different bugs and potentially figure this out.
It'd be interesting to smell if uric acid competed.
Yeah, it would be. And it's interesting too that, specifically monosodium urate, which crystallizes, is what was shown to bind to collect 12A, and then that plasmodial product, hemozoin, is also a crystalline-like structure. And so it makes me wonder if this is going to be like a really glycosylated protein, a lot of sugars, a lot of different linkages that might trigger a collect 12A binding event.
I was also curious, with your experiment that the physicians hate, it does kind of have me scratch my head a bit as well. Did you ever check Aphrodintium for its sensitivity to either gent or the
Cephalothin?
Yeah.
I did not check like an in vitro sensitivity. We did do sequencing, and I can say when animals were treated with cephalothin, Ephrod was gone. So it did seem to be sensitive, at least from 16S sequencing. One in the back.
All right, got a lot of science questions.
TK, all right.
About, so you've done a lot of amazing work with the PhD on really cool stuff. You go back to the beginning. What would you tell yourself?
I should have known you were going to ask me that. Not to be scared. You know, I mentioned my buddy Ricky, and I think I spent a lot of time in graduate school like being scared how to learn things, you know, and I have to give a big shout to my lab who had a lot of patience in teaching me how to do things and sitting with me and holding my hand. But I probably would have said to buck up and stop being a wuss and like, just do it. You know what I mean? I think that would probably be the biggest advice to my former self or any other graduate student that's about to start their journey. Any other questions? Go ahead.
Yeah, so you looked at Aphrodintium in exacerbating colitis model. Have you also tested any other gram positives or gram negatives to see?
No, it's a fair question. And I think it would probably be a good thing to go back to that 16S data that we identified Aphrodintium from and maybe find some of the organisms that were reduced in the collect 12A knockout mouse and see if they could protect from disease. But, you know, only time to do so much. But yeah, that's a good point. All right, so I have bets with multiple people that I'm not going to cry. So I'm going to make this pretty quick. I want to give a shout out to my lab. I gave this talk a week ago in my lab and it was awful. I hope it was a little bit better today, but my lab really helped me hone this in, and they are always your biggest champion and always great at picking you up. And so I just want to give a big shout out to them for their leadership and their kindness, and really couldn't do it without you. Zach, thanks so much for everything over the years. I really appreciate it, man. Manny, Emmanuel Victor, he's only been in the lab just a few months, but man, I've learned so much from him. And my only regret is that he didn't get here sooner, that I could learn more from him. I'm really jealous of our new graduate students that are gonna have the opportunity to learn from him. That's very exciting. Round Lab alumni, big shout out. I've been here a long time, so I've seen some people come and go. The O'Connell Lab, hey, Kaylin, I don't know who else was going to get here with me at 5 o'clock in the morning and kill as many mice as you did. So thank you so much for all of those early mornings and help with all of my experiments. I can't thank you enough. Morgan and I have a podcast. I hope you guys will all check it out. It's called Science on the Side. It's on all your favorite streaming platforms. We even have a few fans that emailed us. It's pretty cool. We talk about science. And Warren Voth, he's not here. I hope he's on Zoom. But you know, cloning doesn't happen in our lab. We're not big cloners. We do a lot of things that we do front to back. Cloning is not one of them, at least not me. And so he has been my god and wizard when it comes to cloning. So I really appreciate him and everything that he's done. My thesis committee, June, you know, she gave me a chance when she probably didn't have to. I was probably a big gamble as a master's student. And in fact, she didn't tell this story, so it allows me to tell this story. I emailed June after going to a conference and learning about the microbiota, and I just emailed professors at the University of Utah working on microbiota. And she came up and I was like, perfect. And I emailed her and she's like, I'm open to having a rotation with you, but I can't find you on our like PhD student list. And I was like, about that. I'm a master's student. And she was like, nope, sorry. And I was like, okay. So I showed up in her office the next morning and I just said, give me a shot. You don't have to pay me. I work for free, you know? And ultimately it worked out. And I think we got some good things done together. And I just really appreciate her for giving me kind of a little bit of a rope to really explore some things over the last few years. Thank you. I really do appreciate it. My sixth members, my committee, David, Matt, Jessica, and Adam, you guys have all been great. Really appreciated our chats over the years. My sixth members, Dennis Wingy. Dennis Wingy was my capstone instructor my very first year of graduate school. And, I was a freshly minted master's student or degree. And I remember going into Dennis's office, having my first draft ready to not waste time and get to it. And Dennis said, whoa, hold on. Tell me about yourself. He was the first one that ever said, tell me about yourself. And I told him my story. I told him that I worked at ARUP and I got this master's degree and I was really interested in science and I wanted to pursue a PhD. And Dennis said, wow, what a great story. And that really meant a lot to me as a mentor, and I've appreciated him and his talks. He wasn't able to be here today. Kiki, Daniel, and Diane have all taken time to chat with me about my project, and I really appreciate that. Path admin, I know there's other people in there. I'm sorry, but Kim, you are everything to us as graduate students, and we really appreciate you. I love you. Thank you so much. You've been a friend and a mother figure, so thank you. Big shout out to my family. Love you guys. Thanks for being here. Probably weird to have your son quit his job and go back to school, but here I am, right? We finished. And then my beautiful fiance, Ashley, I love you. Thank you so much. Ashley and I have been around the world together, but we have not been to North Carolina, and I'm very much looking forward to that experience. So thank you everybody for being here. Hope you have a great day.